this one. Very cool. So we have 2 slides in total this time. So first one is the waste disposal, and the second one is the daily practice. So let's just go through the waste disposal guideline first briefly. Okay. So first, I've separated the waste disposal into 4 categories: the biohazard, chemical hazard, universal waste, and the non-hazardous waste. So just for your reference, and you can click it to go to any site you want. And I also created a flow chart for reference. So you can see that in the biohazard waste, there is separated into the liquid, sharp, solid, and also I'll briefly talk about the animal waste. And also for the chemical hazardous, there are a total of 4 groups: the liquid, sharps, solid, and lab trash. For the non-hazardous, there is glass and plastic only. And also there is the food and drink waste. Be sure not to forget this one. But before I talk about any detailed disposal guidelines, I want to remind 3 of the most asked questions and most dismissed point in our lab practice. So the first one is biohazard waste. The definition of biohazard waste is not only anything that is contact with cells or cell culture things. It is also anything that is contacted with genetically modified materials, which means it is included with anything that is connected with contact with DNA and RNA. And also for guanidine thiosilate, that's very common in the RNA extraction kit, for example, the triazole and RLT buffer. Be sure not to mix it with bleach because it will create a toxic gas. This is very important. And also the third one is the nucleic acid gels that we are using very often. They are treated as the chemical liquid waste actually. So we need to double bag them in the transparent plastic bag and also schedule for pickup. So do not just like place them around in the table and let them bake. Okay. So first is the biohazard waste. I'm pretty sure you guys already are very familiar with all the liquid waste, solid waste, and sharp waste disposal procedure. But I want to highlight that for the biohazard liquid waste, which is the one that we treat with the aspirator and into the flask, please everyone, every time after using the vacuum flask in the biosafety cabinet, run a small amount of 10% bleach through the aspirator. It can not only disinfect the aspirator, but also like put a little bit bleach into the vacuum to disinfect that. And again, do not mix guanidine thiosilate with bleach. Schedule it for pickup instead. For the sharp waste, there's not too much things to say, but just notice that besides the biohazard label that is already on the sharp waste container, it also needs to be labeled of our address, is our room and the UCSD campus. And also this container is a one-time use container, so do not try to recycle them. When it is filled, just take them down into the waste container shed and buy a new one from the UCSD chemical stock room. For the solid waste, the definition of the solid waste is not only those petri dishes and culture flasks. It also includes any tubes that contain less than 15 milliliters of the liquid. So if you have a liquid that contains like small amount of volume of liquids, a tube that contains a small amount of liquid, feel safe to just dispose them into the solid waste, which is this one. Yeah, and also for the pipette tips that we are using on our tabletop and the bench, it needs to go into a secondary container, which is this one we are currently using, and the longer one for the pipettes. And also for the liquid filled, small amount of liquid filled tubes and vials, for example, the 1.5 ml tubes and the 2 ml tubes, they need to be placed into the red bag. Not sure if you can see that. Yeah, so this small red bag, and this red bag need to be placed into this secondary container. And when they are filled, you only need to dispose this small tabletop red bag into the larger solid waste bag. And do I make it clear that how to treat the biohazard contaminated tips and the tubes? Because actually we have some problems about like how to properly dispose of the biohazard tips. So please make sure that they are in a secondary container, and also they are disposed into the red bag. And also the animal waste, I know we don't have a lot of experience how to deal with the animal waste and pathological waste. Just want to stress that it have to be placed into a clear colorless bag, and also into a secondary container like this one. It needs to be stored in the refrigerator or the freezer for no more than 7 days. For pickup, you can contact the ChemCycle or Regulon. So you can either email them or call them to schedule pickup. So now let's go to the hazardous chemical waste. First, I want to make a definition of the difference between the hazardous and extremely hazardous chemicals. So why it is important to make them different is that the empty containers that hold the extremely hazardous chemicals have to be disposed as hazardous waste, but the empty containers that hold the hazardous chemicals can be disposed as non-hazardous glass or plastic waste. So that's why they need to be differentiated. And for how to find them, you can either see this link for a complete list, or you can just go to the UCSD website and search for the extremely hazardous or the hazardous waste. And also how to schedule for the chemical waste pickup, we are using the online tag program. So for how to use it, you can go to the online waste tag program website, and I also link the tutorial video. But another thing that you need to notice is that you need to attach the completed hazardous waste tag to the container before you begin to collect the waste. And also, it shouldn't be in the lab for more than 90 days. So if it is collected for about 80 days, it will automatically be scheduled for pickup. So for the chemical waste, it is a little bit complicated because it has so many classifications. You can see like it is classified as liquid waste, dry waste, lab trash, sharp and piercing objects, including the pipette and pipette tips, and also the empty containers. So for each of them, I have like a detailed instruction of how to label them, what's the definition, and how to dispose of them. But just make sure do not mix the liquid waste with the solid waste. So for example, I saw some waste bottles that have like both the liquid waste and some pipette tips inside. So the pipette tips should be separated from the liquid waste. So about that, so after talking about the classification, there's also another tricky thing about the disposal of chemical waste. It's about those non-hazardous pipette and pipette tips. So according to the regulation of UCSD, they can either be disposed into the non-hazardous plastic waste or the glass and plastic waste. So for example, it can either go to this trash bin or this one. But I think for us, just for convenience, we can just use the yellow labeled glass and plastic waste container, just for convenience. Okay, so so that concluded about all the things that we need to notice about the chemical waste. I know it is the most complicated ways to deal with. So if you have questions, you can always refer back to the slides before, or you can ask me or Douglas for help. So there's also another classification that is called the universal waste, where it includes the batteries, light bulbs, and aerosol cans. So for those ones, you cannot just directly dispose them into the trash. Make sure you bag them up, schedule for pickup. And the detailed instructions for how to use the online tag program is already here. So the last one is the non-hazardous waste. Mostly it is differentiated into the trash and the recycling waste. So for trash, it includes the gloves, weight balls, and the clean wipes. For the recycling waste, it includes the foils, paper boxes, and also the plastic waste. And please be careful that food and drink waste are prohibited in these trash bins. So do not throw any of that into those trash bins. And for the food and drink waste, we have designated small trash bins inside the clean area that is labeled of this clean area label. And for those food and drink trash bins, no gloves or bioagents or chemicals should be presented. So that's actually everything about that we currently need to notice for the waste disposal. So here I linked some of the UCSD websites if you want to know more information about this, and I also linked the contact information of Kenneth and Douglas. So if you have any questions, you can either ask them or let me know. So about the waste disposal guideline, do you have any questions?